wait for a couple moments for the people to join, and we'll start. Okay, I think I can start now. Okay, welcome to week 4 of Bioinformatics Algorithms and Applications. In the last few classes, we've talked about different types of protein sequences, sequence alignment algorithms, and basically what kind of things that you can infer from sequence alignment. Today, we'll be delving a little bit more on the uh, uh, the amino acid properties that confer to the protein. So basically, if uh, how the amino acids are arranged inside the protein to confer specific properties of the amino acids to the proteins. We'll talk about hydrophobicity. Uh, we'll talk about uh, uh, different types of amino acids in terms of the chemical properties and how they are present in different secondary structures of the proteins. And finally, we'll talk about um, sequence alignment properties like constructing phylogenetic trees from sequence alignment and so on. And we'll discuss all those things in due course. So welcome to week 4. Uh, so let's get started. So first thing is in terms of when you're looking at a protein is basically what are the amino acids it is composed of, right? Uh, there are two things that we are interested in. First is the occurrence of the amino acid, like just the number of amino acid uh, in in uh, as many positions in the protein possible. So, for example, if you have a length of the protein to be 200 polypeptides, 200 uh, polypeptide length. Now, out of that, five are, say, for example, alanine, five are tyrosine, 10 are tryptophan, and so on and so forth. So, just the raw numbers is basically denoting the occurrence of the particular amino acid in the protein, and composition a little bit more a little less biased because so for example, if you have a very long protein with a very large number of amino acids in the protein, it's likely that you're going to find a particular amino acid occurring more times in that protein compared to other smaller proteins of lesser size, right? So to normalize the length bias, length of the protein. Uh, so to normalize that bias, we just divide it by the number of residues, total number of residues inside the protein, and we calculate a percentage, right? So basically, now you'll say that 5% of the protein is covered by alanine instead of just saying five uh, times alanine occurs in that protein, right? So again, it tells you when you say a particular amino acid is present in this much percentage of the protein, it will tell you what kind of behavior that protein might be showing, because depending on the behavior of the amino acid, and if the occurrence of the amino acid is very high or very low, it will confer the same chemical properties to the protein itself. So for example, if you have a very high number of hydrophobic residues inside a protein and a very low number of hydrophilic and other polar charged molecules, charged amino acids in the protein, it tells you that this protein might be in that region of the cell uh, where it is not facing any water molecules. So, for example, membrane protein. So you'll have a small hydrophilic head and long tail of hydrocarbons inside your tail part of your membrane proteins. So basically, the hydrophobic part always remains inside, and it is not exposed to any kind of polar molecules or water, and the hydrophilic head will take care of the polar molecules, right? So the composition of the protein in terms of its amino acids will tell you what kind of properties this particular protein might have, right? Now, this is basically a method into uh, how the composition can tell you some things, or how actually the composition is calculated. So, for example, if you have a protein, say P, which you are studying, and Q is the background sample. So the background sample is basically you have a known protein that you are trying to compare against or trying to see differences against. Right. And if you consider PK and QK as the probabilities of observing amino acid K in these two samples, and you assume that the amino acid compositions of the two samples are independent and identically distributed. So these are called IIDs, independent and identically distributed, generated by separate stochastic processes. Like, you can assume these are like random processes that are happening. Yeah. And you have two different probability distributions of different amino acids that can compose this particular set of proteins. Right. The probability distributions P and Q are estimated by computing means and confidence intervals. We'll talk about confidence intervals and in the further slides. Of relative frequencies of residues observed under a set of pseudo-replicate data sets obtained by a bootstrap sampling of whole proteins in the original samples P and Q. So the idea is that if you have 20 amino acids that are present inside your body. So what is the probability that a particular amino acid occurs in this particular region of a protein, depending on the background data that you have, because you'll have a lot of proteins and the sequences, and you'll know at which positions a particular amino acid is occurring or not. So you have those probabilities uh, already known. Right now, if you set a bootstrap, like you randomly sample from those set of distributions at multiple times and take kind of a average out of all your random samplings, it will tell you basically the 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 frequency at which a particular amino acid can occur. What is the probability of that amino acid occurring in that particular position, right? So then what you do is uh, uh, basically define a fractional difference, basically difference in probabilities, in terms of protein P and Q, and basically what is the difference between these two distributions and across different amino acids. So this is basically a simple difference. So at position K, what is the probability of a particular, uh, uh, no, not at position K, at that particular position, whatever position you are interested in, what is the probability that it is an alanine, what is the probability it is an arginine, and so on and so forth. So for each amino acid, you calculate the probability in P minus in probability in Q divided by Q itself. So you get this H parameter which you plot. So it looks like a first set of residues that you are seeing is basically uh, occurring less frequently than you would have expected, except a particular residue, and the rest of them are more or less randomly distributed. So this is what it is telling you, is basically that these set of amino acids are occurring less frequently in sorry, this set of amino acid are occurring less frequently in in P compared to Q. So why is that? Because there might be this set of amino acids might be conferring a particular property to the protein Q, which is not present in protein P, or vice versa. That can happen. So basically, not to make it too complicated, when you look at the composition and you do statistical tests on the composition of different amino acid, what you can predict is basically what is the probability that you will see a particular amino acid at that position in your protein family of proteins, and what kind of function that particular set of amino acid residues is probably conferring to the protein itself. So if you know in protein Q that this set of amino acids are in the active site of the protein Q, that it is possible that that active site is missing in protein P, or that active site is completely changed, or a different active site is present in protein P. So these kind of comparisons are possible with respect to composition of amino acids. Now, as I was talking about different, sorry, is anyone saying?
I was curious to know. So the probability depends upon the type of protein. If we take a globular protein and we
Yes,
compare,
definitely, definitely.
we have
So
to compare
that is why I was
over.
talking about the family of proteins. So if you have if you are taking sampling proteins from a similar family, the frequent probability of a particular residue occurring at a particular position is higher in that particular family compared to other families. Yeah.
Okay.
Okay. Okay, we were talking about different properties that particular amino acids confer to proteins. So one of the major properties is hydrophobicity. So it's a very basic term. What it means is basically hydro means water, phobic is the fear of something. So here hydrophobic means the fear of water, right? So these are the set of amino acids which do not like water. They will not solve it. They will not get uh, uh, form a solution with water. You need organic solvents and other kind of solvents to actually um, mix them, and so basically they do not like water. So mm, uh, it's and it is one of the major driving forces behind protein folding. So if you imagine there are residues which are present inside your in your sequence, which are hydrophobic in nature, so you would like to keep those residues inside the protein as much as possible, much away from the surface of the protein where be it can be uh, like interacting with water, which it does not want, which it won't do. So for that reason, you would want your hydrophobic, or like nature would want their hydrophobic amino acids to be tucked away inside the protein where it is far away from the polar molecules or water molecules and so on and so forth. So generally what happens is that you have hydrophobic amino acids buried in the core of the protein. You have charged amino acids which are mostly present on the surface because they are going to interact with water and other charged molecules, and polar, which are polar amino acids, which are also present on the surface, or it helps in hydrogen bonding with different. So a protein might not be a monomer; it can be dimers, trimers, tetramers, and so on and so forth. So hydrogen bonding is a very important bond that keeps the protein in its place in its tertiary and quaternary structures. So these kind of polar amino acids help in that. And one thing to note is like every amino acid is hydrophobic to a certain extent. So it is not a binary thing that it is either hydrophobic or not hydrophobic. It is a spectrum. So some amino acids are much more hydrophobic than the others. Some are much lesser. So just to keep that in mind. Two examples would be isoleucine and valine, which are strongly hydrophobic, and arginine, which is hydrophilic. So these are your non-polar uh, hydrophobic. So isoleucine and leucine, you can see long hydrocarbon chains, which give them this property of hydrophobic nature. And you have, say, for example, uh, arginine, which is a charged amino acid, which is going to be hydrophilic in nature, and basically, and since this is charged, it can interact with water molecules very well. So that is good. Now, how do you measure hydrophobicity? Right? It's a you just say it's hydrophobic or hydrophilic in nature. It's a qualitative trait that you're describing, right? But how much hydrophobic is it, or how much hydrophilic is it? We need some scales to tell that, right? So there can be different types of scales. One would be finding experimentally, right? So this is one paper which is like kind of uh, the basic natures of so it describes amino acid in different in terms of different hydrophobic scales. So this is one such paper where what they've done is basically done experimental conditions in terms of like how much solubility is there in ethanol, how much solubility is there in water, other organic solvents, and so on and so forth. And they've experimentally found out which molecules, which proteins are sorry, which amino acids are more hydrophobic compared to others, and so on and so forth. And they have made a scale out of that, and different values they have conferred. This is a quite an interesting paper to read. So if you're interested, I definitely uh, ask you to go and take a look at this. So these are experimental procedures, but by which you can quantify hydrophobicity. But there are computational procedures as well. Uh, one such as basically, I think and one is named after Professor Michael as well. So there is one for there as well. So I'll not be discussing that, but this is another scale, which is the kite Doolittle scale. So this is based on where the residues are found in the crystalline structures. So if you do not have crystalline structures of a protein, this kind of like the more and more crystalline structures you have of proteins, the better the scores are can be derived. And if you do not have many, say for example, you're looking for an amino acid which is present in a very terminal end of a protein, and you're trying to find out what property it is conferring, but you do not have enough crystalline structures of the proteins of which this is amino acid is part of in the terminal end. So in those kind of situations, these kind of scales are not very informative because you do not have the data itself. But obviously you can do a experimental procedure, find the amino acid, make it a terminal amino acid, and then um, study how it is all how it is getting mixed in water, in ethanol, in other organic solvents, and so on and so forth. Right? But yeah, this is a very widely used scale, uh, and yeah, this is actually based on where the amino acid is in the 3D structure in crystal extracrystallography, right? So, yeah, the scale tells you that regions with a positive value are hydrophobic, and basically negative values are hydrophilic. And it can be used to identify both surface exposed regions as well as transmembrane regions. We'll talk about transmembrane protein a little bit later, depending on the window size used. Now, the window size is an important concept here, right? So, why do you need a window size? So, again, if you are folding a protein, and if you, with the assumption we are going that these set of hydrophobic amino acids will be tucked inside the folds and away from the surface of the protein. So, right. So you need a residue, a series of residue of hydrophobic proteins near the folding that is happening in the protein, right? Because you need not a single amino acid, but a set of amino acids, right? So for that, when you calculate hydrophobicity in terms of the folding, you calculate in terms of the set of residues, not a single residue. So for that, you use a sliding window, not like a single uh, amino acid residue, right? So we generally use window sizes of 5 to 7 uh, for predicting surface exposed regions. So when it's a charged amino acid present in the surface, it will generally be of length like 5 to 7 residues. But when it is inside and it's very hydrophobic in nature, there you need 19 to 20 residues around to fine transmembrane domains, right? Uh, Again, these values should not be used as rule of thumb, and the deviations might occur depending on various factors. But generally, the idea is that you have a score like this, which can tell you relatively. This is again a relative score, depending on the score. It can tell you how much hydrophobic or not hydrophobic a particular amino acid is. So this is the set of Chi2 little scores. There are other scores as well. I'm not going to go into this, but I found this. I'm just going to give you the link to this. I think hydrophobicity scales. Yeah, I think this one. Yeah, here they describe all the different scales that are possible, and they are giving uh, the basically the publications associated with it. This is the Doolittle uh, paper, which is the Kai Doolittle. mm, Yeah, so this is the paper. I don't think this is freely available, but I think you can get this paper. This is a very old paper. I'll give a link to this. Maybe you can take a look at this article as well. Okay, so moving forward, so we know hydrophobicity is conferring, like, so there are a set of amino acids which can give this hydrophobic uh, properties to the protein, and mostly these hydrophobic residues are present inside the folding regions, and all your polar and charged amino acids will be present in your surface level. But again, inside the set of residues that are responsible for giving this hydrophobic nature to the protein and are present inside the folding, and these are not a single amino acid, these are set of amino acid residues depending on what type of protein it is. If it's a transmembrane protein, you'll have a lot higher. If it is like a normal protein, you might be okay with a little less number of amino acids in hydrophobic amino acids in the fold. Okay, okay. So again, this is the explanation for the sliding window and hydropathy plot. So all these things, this set of values and everything. Now, if you plot these values across all the sequence, so if you consider the x-axis to be the sequence, and each of this line actually corresponding to a particular amino acid, and then you plot the hydrophobicity values for these amino acids, and then you see some peaks, some troughs, and so on and so forth, right? So this signature, but there is a lot of hydrophobic amino acids in this particular region, can tell you that probably this is a region where the fold is occurring, right? Uh, and so in many cases, you'll see that if you are trying to predict transmembrane helix, because it is a complicated structure, because there are a lot of folds, so like it folds 2 to 3 times, and depending on the protein itself, the folding can be much higher. I think GPCRs are an examples for this, and they have, I think, if I'm not incorrect, they have, I think, 7 folds. So this kind of folding nature makes the makes a protein very interesting to study, right? So you plot this hydropathic plot. It's nothing. It's according to the sequence you are plotting what are the hydrophobic values from a particular scale, right? There are many hydrophobicity scales, as I said. Some might be computed from experimental solutions, study of free energy transfer. from aqueous solution that mimics a membrane. Some use crystallographic data like the Kite-Doolittle one. Right? Looking at individual amino acids creates a noisy graph because hydrophobic and hydrophilic residues alternate frequently. So to find meaningful structures, we use a sliding window and we use a formula something like this. So basically, along a window, you sum up the hydrophobic values for a particular uh, for a particular region. So this is the region which is I minus k to I plus k. So if you consider the kth amino acid, I residues no sorry if you consider the ith amino acid, so if you consider the ith amino acid, so your sliding window is from I minus k to the left and I plus k to the right. So that is the window that you are taking. So generally, this is like 19 to 20 residues, as I was saying. But yeah, the idea is that if you just do like a very small set of amino acids, 2 to 3 to 4, you will not get an idea what is actually happening. So this is like the resolution is not a single amino acid. The resolution is based on this the value of k. How many amino acids are you going to take? 10 upstream, 10 downstream. I think that should be fine. So probably these are like corresponding to 20 amino acids at in the sequence. Right? And this tells you the signature will tell you possibly there is a loop, there is a helix, or and so on and so forth. Right? So I wanted to show one example of this. I think this is open in my browser, but this is the ProtScale database. So if you mm, give a UniProt ID, so this ID is for a glycophorin protein. This is a transmembrane protein. And if I just wanted to see the hydropathic
So which protein?
human glycophorin, so let
Okay,
me
okay.
UniProt. Yeah, this is the protein, the first one, right? Uh,
Okay, sir.
yeah, and so I've just put the UniProt ID here, and if I just look at the hydropathicity with respect to Kite and Doolittle, I do a submit. Now it will tell me these are the domains or present in this protein. So this is the helical domain. You have the extracellular domain, you have the cytoplasmic domain, and so on and so forth. So the helical domain will be the hydrophobic one, right? So and the extracellular are the one which should be hydrophilic. So if you go to the extracellular part, this is basically the whole sequence, and this is the fragmentation that they're considering. If you just want the faster sequence, it is given like this. It's 57 amino acids, and you see this is the hydrophobic scale of Kite Doolittle they're using, and these are the scores, right? Now you see the scores are ranging from -2 to positive zero point 0.5, so it's fine. But if you compare this against, I go back here, against the helical region, which is um, the hydrophobic region, then you see the values ranging from 2.6 to 3.5. So the amino acids present in this particular region of the protein, for this case, uh, uh, transmembrane helical protein, which is glycophorin, inside in the transmembrane part where hydrophobic residues should be there, you see a very high score of these hydrophobic residues, so ranging from 2.6 to 3.5. There it was like -2 to 0.5. So the scale is much different here. If you look at other regions, say cytoplasmic, here also the scale will be not very high because you need charged or polar amino acids in this set of regions. But in the in the folding region, the transmembrane region, you need hydrophobic amino acids, so there the scores are very high. So you can try with different proteins. It's a very simple thing to do. You just go to UniProt, you type in any protein. Let's I don't know if UPCR will come or not. Yeah. So if I give something like this is also another, no, this is, yeah, I think this is the one that you should use. So if you take this, Yeah, so this is a GPCR protein also. This is also transmembrane receptor. So you see extracellular, you go, the scale is this, so maximum is 1. Now if you go to the helical sides, you see it's ranging from 2 to 2.9. So you can try with a few proteins and see. It is quite interesting to see actually which part of the protein is actually the hydrophobic part, and if it is hydrophobic part, for most cases it is embedded within the membrane and so on and so forth, right? So this is an easy thing to do, and these are like different domains that that this particular protein has. Okay, so coming back here. So as I said, why we are using this sliding window? Two reasons. One is like there is it's not a responsibility of a single amino acid to confer that hydrophobicity. It is done over a set of amino acids, maybe 20 amino acids around the region that it is folding. And the second thing is like it is not imperative that all the 20 amino acid in the protein folding region must be hydrophobic. There might be alternatives. So one hydrophobic, then hydrophilic, again hydrophilic, and so on and so forth. That can also be present with a more density of the hydrophobic amino acid. Right? So to do that, we use the sliding window concept where we just sum over. If you take one amino acid, we take amino acids that are 10 amino acids upstream, 10 amino acids downstream, and we calculate it for a region, not a particular amino acid, right? Now, the reason is I'll talk a little bit more on the reason why this is happening is because there is, if you see the secondary structures, the alpha helices, the beta sheets, and so on and so forth, uh, you have a lot of amphipathicity. So amphipathicity means like both hydrophobic and hydrophilic properties there. So the alpha helix is the most common type of secondary helix structure. They're formed when the carbonyl O of the ith amino acid forms hydrogen bond to the amide H of the I plus fourth amino acid. So four amino acids away, there is a hydrogen bond forming, which gives the helix its structure, right? So this is actually, move this here. Yeah. So this is how the structure looks like. So this is the I plus fourth amino acid, and I think this is the ith amino acid, and they form this particular helical structure. So this is I plus one, I plus two, I plus three, and I plus four. The I plus fourth amino acid will form a hydrogen bond with the ith amino acid. Right. Uh, so this is an alpha helix for this particular sequence, and yeah, so I think this is showing the ith amino acid as acetate and I plus fourth amino acid as serine, right? So I think that is good. Okay, now this is how the structure will look like. Right? So the ith amino acid will form hydrogen bonds with the I plus fourth amino acid. Now the whole set of amino acid chains in the R groups, they can be either hydrophilic or hydrophobic. So if it's a charged group present, it's probably hydrophilic. If it's a long hydrocarbon change, it is probably hydrophobic, right? So they can be localized in specific positions on the helix, forming these amphipathic regions. So there can be, so these regions in the secondary structure are conferring to both hydrophobic and hydrophilic nature of these amino acids, right? So yeah, they're localized in specific positions in the helix, forming these amphipathic regions on the protein, or fully hydrophobic helices may extend through the plasma membrane as well. So in amphipathic helices, where you have both hydrophobic and hydrophilic residues. The hydrophilic residues are positioned on one side of the helix. So it's a helix, like it's a helical structure. It's a ladder-like structure if you twist it like so. There will be amino acids on one side. There will be amino acids on the other side, right? So in one side it is going to be hydrophilic, and the hydrophobic residues will be present on the other side. And the R groups may entirely also be hydrophobic within the alpha helices that span the plasma membrane. And helical wheel projections show the polarity of the helix faces looking down the axis. So if this is basically a helical structure, but you have to imagine that you are seeing this structure from top down. So all these actually amino acids are part of the helix, and you are seeing it from top down, right? So, and on one side of the helix you have your non-polar amino acids, and on one side you have your polar amino acids. So yeah, this is a top-down view. Like, so this is amphiphilic in nature or amphipathic in nature. Right? Just a second. So this is an example of the human receptor type tyrosine protein phosphatase C, which is this is the it's basically the region that it is showing, and you see this is a very hydrophobic heavy region of the protein, because this is a membrane crossing protein. You need these hydrophobic residues to be present if your protein is basically passing through the membrane, because the inside of the cell membrane is also already hydrophobic, right? So both these kind of things can happen. So there can be localized hydrophobic and hydrophilic nature of these amino acids that can be fully conferred by hydrophobic amino acids, or vice versa, fully conferred by hydrophilic amino acids. So different types of combinations can be possible is what I'm trying to highlight here. Right. So take another example. I think this has also been discussed in the class. So it's a beta barrel protein, right? So you have transmembrane beta barrel proteins, mostly found in bacterial outer membranes, and they are harder to predict than helices because they don't have this long run of hydrophobic residues, right? Instead, what they have is an alternating pattern: hydrophobic, then polar, then hydrophobic, then polar, and so on and so forth. So it's an alternating pattern, right? So uh, uh, how do you identify that these are the set of beta barrel proteins? You go back and see the composition of the protein, as I was discussing in the first slide. So you identify them by analyzing the composition bias. In this case, you'll have very high serine glycine residues and specific motifs that are present in this protein. Now, the beta strands inside the barrel proteins are amphipathic in nature. So one side faces the lipid, the other side faces the water-filled pore. This creates this alternating pattern, which is hydrophobic, polar, like this. Now, when you calculate the hydrophobicity, you calculate while skipping one residue, right? So when you do that, you do it like this: I minus 2, I plus 2, I plus 4, I plus 6, I plus 8, and so on and so forth. It is skipping every other residue because alternatively, there are hydrophilic residues present, and there is no point in calculating those things. Right? So this calculates the average hydrophobicity for one phase of the strand, because the other phase, which is composed of hydrophilic residues, are facing the water pore. Right. So if you want to calculate the hydrophobicity of a particular region of this protein, which is facing the lipid, basically you have to do it using alternate amino acids, because uh, the alternate amino acids are going to be present, the consecutive amino acids are going to be present on the other side. Okay, so yeah, we talked about hydrophobicity scales and how different values of hydrophobicity can tell you what region of the protein it is, if it is folding or not, and so on and so forth. Right? Uh, one in, Another important thing to talk about is basically patterns of these sequences, right? So functional sites in proteins, like in enzymes, the active sites.
Sir, I have a question
Yeah, sure.
regarding I have a question regarding hydrophobicity application. So we are finding hydrophobic characteristic of a protein or a
Sorry,
part
I
of a
can't
protein.
hear you. Can you be a little louder?
Sir, can you speak me? Can you hear me now?
Yeah, 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 I can hear you.
Sir, I was asking what are the applications? to know the hydrophobic character. So we are finding the hydrophobic character. So why are we doing that?
So mostly the application is to find where the protein is folding. So you're given a sequence of amino acid, and all you know is just the sequence and the composition of the protein, right? So you have, say, for example, a protein is made up of 200 amino acids. You know the sequence of the amino acids and what amino acids are present in the sequence. This is all information that you have. Now, if you're trying to predict what kind of protein it is, right, you can do it in two ways. One is already have a set of known proteins. You do alignment of this protein to the known proteins and try to find out if they are from the same family or not. Or what you can do is try to predict what kind of folding that can happen in your novel protein. So if you are finding this set of hydrophobic residues in certain regions of this protein, say you are finding it in five different regions with very high peaks, the plots that I showed, you are finding in five different regions of very high peaks, which means that these are the regions where the folding can occur. So if you are finding these five different regions where folding can occur, so there's a possibility that this protein can fold five times in an opposite way, which can in from which you can infer that this might be a transmembrane protein because it is folding so many times, right? So these kind of inferences mostly about the structure of the protein, and also it can also tell you about the active site of the protein. So if you're if you're, say, for example, studying an enzyme, and that enzyme binds to, um, say, for example, some hormone, right? So, some compound inside your body which is charged, right? Now the set of amino acids that will bind to the to the substrate will have to be charged also, right? So you will have to find a region in the sequence where you have a very low level of hydrophobicity, and mostly dominated by the serine, threonine, glycine residues, because these are the residues that are going to be that generally bind to different substrates. So what I'm trying to say is basically, depending on the composition and the hydrophobic nature of the amino acids, you can predict what kind of protein it is. Also, you can predict to a certain extent, not fully, obviously, maybe to a certain extent, how the folding of the secondary structure, at least not the tertiary or the quaternary, at least how the secondary structure might look like. So mostly these applications, and like if you have a lot of hydrophobic residues inside a protein, you might say that this protein is present in the cell in a place where you do not have enough water molecules. So if it's a protein that is completely embedded within the membrane, you'll find a huge number of hydrophobic residues compared to the number of hydrophilic residues, and then you can make a inference that
Okay.
this is present in a place where there is less of water, more of liquid, something like that. Yeah.
Okay, sir. Thank you, sir.
Yeah. Okay. Just then give me a minute. I'll just be back.
Yes, sir. Yes, sir, you are.
Mm, yeah. Am I audible now?
Yes, sir.
Yeah, it's fine. Okay. Let's go back here. Yeah. Yeah. So yeah. So we talked about hydrophobicity in terms of the sequence of the amino acid. Now, when you're trying to find a sequence of the amino acid of a set of amino acids in a protein, the idea is that some regions of the protein is well conserved in terms of the family of proteins it belongs to. So, for example, if you take all the transmembrane proteins in the human body, and then you look at this sequence, some regions of those proteins will be conserved across all the transmembrane proteins present in our body or in our cell, right? So, it is possible that uh, you are suspecting when you're doing a, say for example, a multiple sequence alignment, you see a set of residues that are getting conserved, but there are a set of residues that are not getting conserved, and like you are trying to infer if that region is a binding site, or is it a hydrophobic set of residues which is helping in the folding, or any other kind of residues that are present in the protein, right? So instead of the exact sequence, what you can do is search for a very uh, kind of redacted kind of sequence. So instead of searching for an exact sequence like this, you use a regular expression which is called regex to define a flexible rule. And this is how the syntax of that looks like. So this is for a particular database called ProSite. So here what it does pattern matching. So if you give a set of patterns, not the full pattern, some like half the pattern or like whatever pattern you have, it can find that pattern in other proteins and give you a result that these are the set of proteins which have a similar set of residues, right? So this is what the regular expressions look like. So you can, these are the different syntax. So if you give something like the AXA, which means like alanine, X can be anything, and then another alanine. There can, you can give an inclusion list. So for example, if you give something like this, it means like at that particular position there can be a leucine, isoleucine, valine, or methionine, or something like that. You can give an exclusion list. At that position it can't be lysine but any other amino acid, and so this is the syntax how it looks like. This is the database. ProSite, you can take a look at it. Like it will give you uh, results based on the pattern that you are providing, and this can help you understand. So if, even if you don't know what kind of region that particular set of amino acids are conferring to, if you find the same set of patterns in, say for example, an active site in an active site of another protein, or like in an active site of like 5 different proteins, there is a high possibility that that region is an active site. So for comparing patterns, you have these databases like ProSite, right? Uh, Now I'll come to this topic of protein position-specific scoring matrices, which are PSMs. Right, these are very important in terms of like multiple sequence alignment and different models that you do, that you make. Even to a certain extent, the scoring matrices that we use for sequence alignment, those are also somewhat based on a similar idea. Yeah. So. So if you just, when we do sequence alignment, so if you remember, we have three categories. We have a match, we have a mismatch, we have a gap, right? But again, if you're just looking at patterns of amino acid residues which are doing the similar kind of functionality, uh, it might not be a one-to-one match. It might not be like you need to have all the same amino acids in that particular region to do the same work. Some variations are possible, and some variations are definitely there in different proteins that we see. Now, that is the thing. The idea is that that binary results match or mismatch is basically very rigid and very harsh, right? So what you do is you use a probabilistic calculation of like if there is a match or a mismatch. So these set of scoring matrices are probabilistic in nature. We build a matrix where the rows are the 20 amino acids and the columns are positions in the sequence. Each cell contains a score representing how conserved that amino acid in the position is in that position. So you use prior data to find how out of 500 amino acid, 500 proteins in 300 proteins you see a particular same amino acid at that particular position. So probability will be 3 by 5 for that position, right? So uh, yeah, and also you add pseudo counts to ensure that no probability is zero. So if you do not have, say, for example, in 500 proteins, you have zero proteins in which you have that amino acid in that location. Instead of using a probability of zero, because this again it'll become binary, right? Zero and one. We do not want to do that. We add a very small value of probability to say that this is basically a pseudo count, right? Pseudo zero. Uh, in a scenario where you have a set of related proteins and then you ask a question if a given sequence is a member of this set or not, you compare it to a consensus sequence. So you have a particular protein and you have a family of proteins, hundreds of proteins there, and you want to say that if uh, is this particular protein that you are interested in is a part of that family or not, right? So what would you do? Generally, the idea would be to have all the 100 proteins and then your protein, the 101th protein, and then you do multiple sequence alignment, right? But just a sec. Yeah, ideally that would be the way to go. But the thing is, each of the sequence between these hundreds of sequences, there is a lot of variation. Not all the positions are conserved. These are not 100 identical sequences, right? So multiple sequence alignment will not tell you much, right? Because it's not one single sequence. It's like hundreds of sequence, and you're trying to match one sequence to hundreds of sequence. So that is not a very good way to say if two sequences are similar or not. So instead, what you do is basically find a consensus sequence. So what is a consensus sequence? It is a sequence where each position is defined by the majority vote based on multiple sequence alignment. So what you do is you take those hundreds of proteins and you do multiple sequence alignment among them and take the majority at each position. So in the first position, all of them are P. Second is all of E. That is fine. In this case, you see in this particular position, three are I's, two S, and one F. So at this position, it is I. So you take the majority according to multiple sequence alignment, and then you do what are known as sequence logos, which are a continuation of the consensus sequence. So uh, yeah. So what is this? Is basically it's a logo which displays the frequencies of the bases at each position. So this is only for nucleotide bases for DNA, but this can be done in proteins as well. So the example is for DNA, right? So if you look at each particular residue, so for example in this one, right? So all are C's. So the length of the C, height of the C written here will tell you the proportion of individual proportion of sequences that carry a C there, right? And if it's a combination of some things, then you have this. So in this particular position, maximum are T. So you have the biggest T, then a few C, two C's, that's C, and there is another G here, which is very low because there is only one occurrence. There is also one A here, which I don't think you can see. So the idea is that the length of the character will tell you what is the occurrence of that character in the set of sequence. And this is basically the article which describes sequence logos. You're going to come across sequence logos in a lot of places. Sorry. Yeah. Yeah, this paper describes what sequence logo is. So this is the person, Thomas Schneider and Michael Stephens. They are the people who actually coined this thing, and like we follow that after that. So you can take a look at this. Okay, okay. Now, the I
Sir, back to the sequence logo. I have a question.
yeah, sure.
Yes, sir. In position number 7, there are all T's, but in the logo it says an I is also present. Not there.
Which position? Tell me. This one.
Uh, the last, yeah, this one.
This is only T, right? Are you talking about this bar?
I, yeah.
That's an error bar. That's an error bar that calculates what is the standard deviation of your. Uh, so so when you are drawing a bar chart, you draw the bar to the level at which the value is. So if you're drawing a bar chart, if your value is at 5, you draw 5 till 5. If the average value is at 5, right? Now, if you have hundreds of data points and the average is 5, you're plotting 5, but with this set of error bars, you're showing the variation in the mean. So this is basically
Okay, sir.
an error
Error.
bar. This is not I, by the way. Yeah, this is an error bar.
Okay, sir. I thought it represented I
No,
solution.
no, no, no.
Okay, okay, sir.
Okay. Uh, yeah. Now, say for example, the same thing. You have sample sequences of a protein family, and you are given a goal. So you have the protein family, which are like same set of proteins of similar functions and similar sequences. Uh, you are Your goal is to provide a stochastic model of the family, a model describing this set of sequences. How do you do that? First is basically you do multiple sequence alignment, as I showed here as well. So if you have protein family, the idea is a family of anything. So the first step would be to do a multiple sequence alignment, and you construct this PSSM, right? Position specific scoring matrix, and the score of the amino acid at A on the position I will depend on the frequency of observing A in this position in the multiple alignment. So basically, this particular alignment, right? Uh, the the considerations that you should keep in mind is basically, should the sequences in the family be weighted equally, uh, and as I was saying, that you have to add small probability values so that the probability is not zero, because probability zero meaning it is going back to the binary thing, right? So we do not want that. We'll add a small probability value, right? So uh, this is the idea. So you do the multiple sequence alignment as done here. You collect frequencies of each amino acid and gaps for gap alignment at each positions, and you compute log odds score for observing a given amino acid in a given position. So what is the basically so if you are if actually I'll say describing the example. So this is how the multiple sequence alignment will look like, and after that you create the PSSM, and basically you add a little bit of pseudo counts to avoid zero probabilities. So that is there. Now, how would you calculate the probabilities? So the point is, you need to have a background idea of like the from the data that you already have. What is the probability of a particular amino acid at this position, and what is the probability of that amino acid in the database? So what you're doing is basically in the database, you're searching for a family of proteins. In that family of proteins, what is the probability that a particular amino acid P occurs at position I? So that is your PI. And the capital P is basically across your database, all the proteins that you have, what is the probability that that particular protein is at position I. So if you get these two probabilities, you take a log odds for that. So you take log pi by P, and that is basically your score for your PSSM. So this is your sequence. So this is your sequence positions. This is your sequence, and so each box will tell you. So at position 1, you have this amino acid P, and what is the log score, uh, the score for that particular uh, amino acid at that position being across the database. So you calculate the probability of that amino acid at that position across your family. So in this particular case, the probability will be 1 because all your uh, um, in across all your proteins in the family, the first position is P, and the capital P will be the probability that that amino acid, that P amino acid, is present in the first position in your database. So, say for example, it is present in 200 out of 5,000 um, proteins in your database. So your this capital P will be 200 by 5,000, which is like 2 by 50, and this will be 1. So this, then you can calculate the log of the ratio, and then that will be, and if you do that for all the sequence, all the amino acids and every sequence, every position, you get what is known as this PSSM. And again, a little bit of like things, nuances that you need to add a pseudo counts, basically like a little bit of probability. I'll not go into details how that is calculated, but yeah, you add a little bit of pseudo counts so that it does not become zero. It's the same as when you are doing log normalization, right? So when you are taking log for all the data points that you have in your data, you add a small variable to that, to your data before taking a log, because if you have zero data, I mean, if you have the value 0 in your data, log will not work because log 0 is not defined, right? So you have to have some positive value to take a log of that. So it's the same case here. Since we are taking log, we do not want the probabilities to be 0, and also it will go back to that binary thing that we don't want. So we add a small probability. And how do you do that? Like, how do you calculate that probability and add that? That's a separate conversation, but like the idea is that it's simple. Okay. Now I'll talk about another part of the course that has been taught to you is phylogenetic analysis. So till here we are comparing sequences, we are comparing profiles of the amino acids in these sequences. Now what we want to do is basically based on this comparison, if there are similarities or differences, we want to quantify that. Right? So till now we are doing sequence analysis. We are seeing how many residues are conserved, how many are not conserved, how many are hydrophobic, how many are hydrophilic, and so on and so forth. So if you are given us hundreds of sequences like this, can you tell me, based on these things, this analysis, sequence analysis, similarity, dissimilarity, how each of these proteins are related to each other? Right? So uh, the phylogenetic analysis is a major part of that, and it's not only done, like it's not only can be done on protein sequences. It can be done in any kind of things. Phylogenetic analysis is a very basic thing to know, but it is very useful in in different types of clustering algorithms. We'll talk a little bit about that as well. Okay, so a phylogenetic tree or a evolutionary tree is the graphical representation of the evolutionary history of biological sequences and allow us to visualize the evolutionary relationships between them. So if you just look at, like, if you don't read that, if you just look at this particular tree, there's a node. These are your branches. So these are your branches, and these are your basically nodes also, but these are, since these are the end nodes, these are known as leaves as well. So this is root, this is branch, and this is leaf, and which is, the leaves are called the terminal taxa. Each of these leaf is known as a taxon, and again, node is basically where the branching is occurring. Sorry, this is a node, right? uh, I think some of you might be familiar with the graph theory. So if you're familiar with graph theory, this is quite, yeah, it's going to be quite easy for you to understand as well. Okay, so now just from looking at this particular diagram, what can you say in terms of the relationship between any two? proteins. So just assume that these are 5 different proteins, and this is a phylogenetic tree that is given. What is the first thing that you can notice? Notice is basically which of the two among all these 5, which is the pair which is the closest, right? In this case, it can be B or C because they are directly connected. It can be D or E, they are also directly connected. Now, the discriminating factor would be the length of these connections. The length of this branch is actually proportional to the evolutionary distance between them. So in this case, I think D and E are closer than B and C, if I'm not wrong. But like this is just a visual um, uh, interpretation that I'm making. But the idea would be that the branch length would be given, and you can calculate the branch length. Calculate the divergence between the any two pair of individuals or proteins using the the length of the branch between them, right? Okay, so evolution explains the diversity of life on Earth, and understanding evolutionary relationships help us understand the origin and relationships between different organisms. Molecular phylogenetics, which is what we are trying to do, is an important aspect of bioinformatics that helps us understand evolutionary history and relationships by using molecular data. In our case, it is mostly protein, but also DNA sequences. But DNA sequences are a little bit more trickier than this. But yeah, the idea is that, right? So just to cover our bases, majorly two type of trees with respect to its structure. It can be rooted, so there is a common ancestor there, and in some cases it is not rooted; it is unrooted, which means there is no common ancestor between all these set of individuals or proteins. Right? Now, one important thing, this is more important for your exam than it is for your understanding, but like the idea is small problems from this can come is basically if you have a rooted tree versus if you have an unrooted tree, and if you have n um, uh, number of proteins or individuals in that tree, what is the total number of rooted trees possible? What is the total number of unrooted trees possible? So these are the notations. So I'm assuming you're familiar with the factorial representation. If not, please look it up. It's just if it is n factorial means n times n minus 1 times n minus 2 times n minus 3 till we go to 1. So that is what the factorial means, but I think we already know that. So for rooted trees, this is the formula, and if you just divide it, it becomes this 2n minus 3 double factorial. The double factorial means that you multiply alternatively. So you do not do n into n minus 1, n minus 2, n minus 3. You don't do it like that. You do n minus 2, n minus 4, n minus 6, and so on till 1. Right? And yeah, same for this case. So for rooted trees, it is 2n minus 3 double factorial. And for unrooted trees, it is 2n minus 5 double factorial. You can derive this using permutation and combination, but I'm not going to go into the derivation, but I hope you get the idea. And like I'll recommend you remember these formula because a small problem can come like this in the exam because this is a very easy problem to give, right? So okay, so on the basis of structure, like if it can be rooted or unrooted based on evolutionary history. Sorry. Yeah, based on topology, it can. There are two types of these phylogenetic trees, which first one is cladogram and the second is phylogram. So the cladogram is basically a type of phylogenetic tree that displays the branching pattern of displays only the branching pattern of the evolutionary relationships among organisms or proteins in our case. Cladograms are unscaled, which means that this branch length will not tell you anything about the uh, sh- uh, amount of evolutionary divergence between taxa. Basically, the length of this branch will not tell you that DNA, how much DNA are separated, and or this branch won't tell you how much B and E are separated. On the other hand, phylogram is basically that tree that will represent the relationship by showing both the branching pattern and also the amount of evolutionary divergence, and these are scaled. So, for example, this divergence is lower than A to C divergence. D to E divergence is going to be lower than A to C divergence. So these scaling will tell you uh, how much in terms of like just it's a qualitative measure, but relative to other divergences. Okay, so, for example, you're comparing between A and C versus D and E, which is higher and which is lower. It can tell you because the branch lengths are scaled based on the evolutionary divergence, right? Okay, now how do you construct this tree, right? So in our case, we are going to look at mostly protein sequences, but you can do it from DNA sequences. Uh, you can do it from a lot of things. Phylogenetic tree you can construct from any type of data, right? Continuous data. So in our case, we select a molecular marker. So in our case, the molecular marker is protein sequence. Then we do multiple sequence alignment across different proteins. And then we select a model for evolution. So basically, this model for evolution is the scoring matrices that we use for alignment. So we talked about this Jute Skander model and Kimura's model. Basically, it is transition transversion. What is the probabilities that you use? In if you are using protein sequences, we mostly use PAM or Blossom. So these are the scoring matrices. Is basically the model of evolution, and then you construct the phylogenetic tree based on the distance between each sequence, right? Based on the score. So this model of evolution will tell you the score at every for every alignment, right? So uh, between a pair of proteins based on that alignment score, you can uh, you can imagine the relative divergence that has happened between these two sequences compared to all the other sequences that you are comparing against, right? So yeah, these distance-based methods rely on computing the amount of dissimilarity between sequences, which we do using multiple sequence alignment or pairwise sequence alignment using a particular scoring matrix, right? Uh, yeah, the amount of dissimilarity between sequences, while character-based methods use molecular sequences from individual taxa to trace the character states of the common ancestor. But in our case, we'll just use sequence alignment and we'll use the score as a proxy to divergence of two proteins, right? And finally, this is important, you assess the reliability of the tree. So how do you assess it? I don't think I have slides for it. Do I have it? No, I don't have it, but the idea is that how do you assess the reliability of a tree? So, if somebody is of statistics or mathematics background, I think you have heard of random sampling and bootstrapping, and how do you calculate confidence in intervals of, say, regression coefficients and so on and so forth. So, basically, the idea is that this is a statistical measure that you are saying that when you're constructing the tree that these are like these sequences have diverged more versus these sequences have diverged less and so on and so forth. All these are based on some statistical measure that you are using. In our cases, it's the alignment scores or some other things that you are using, right? Now, these can be biased. This can be a little bit misleading at times. So what you do is you randomly bootstrap this, right? So uh, we use this method called bootstrapping, which is basically you repeatedly resample. So you have set of 100 samples. You take 80 samples out. You calculate your test statistic, and then you put those 80 samples back. Then you randomly take again a set of 80 samples, calculate your test statistic, and you put those 80 samples back. Now you do this for hundreds of times, and hundreds of times to calculate the statistic. The idea is how much variation is there in the statistic. If your model is robust, if your data is good, your assumptions are fine, then you won't see much variation in this test statistic across so many bootstrapping levels. But if your model is not robust, if it is not fully correct, you'll see a lot of variation, and that will create a very high confidence interval level. Right, so that is not a very good sign of a good model. You want your confidence interval level to be very narrow, very small, right? So that is how you assess. So you bootstrap, and you resample, and then you see, is the relationship completely getting changed or is it not changed? So that is how you assess the reliability of the tree. So if you are a little bit confused, I'll suggest you take a look at resampling. You take a little bit of look at bootstrapping. Bootstrapping is a very commonly used method across different statistical measures that we use, right? So yeah. Okay. Now you have, say for example, gotten uh, this set of sequences. Now, based on distances, you are trying to construct this tree, right? So assume that for any pair of species, we have an estimation of evolutionary distance between them. So, for example, alignment score that I was talking about, and your goal is to construct a tree which best approximates this distance. Sorry. Now, how do you construct a tree from a distance matrix? So the idea is that so first you create a matrix, right? So this is again going a little into graph theory. So if you are, this is a graph structure, right? So this is a graph structure where each of these are basically your so one is a root, other are leaves, there are nodes, there are branches. So basically this is a graph notation when you have where you have a set of vertices and a set of edges connecting these vertices, right? And how we describe a graph notation is through something called an adjacency matrix, which very much looks like the matrix on the left here. So what it tells you is basically A and B are connected using weight 2, A and C are connected using weight 7, A and D are connected using weight 7, A and E are connected using weight 12, and so on and so forth. So this is what that adjacency matrix looks like. So yeah, it's an n by n matrix where n is the number of samples or the number of protein sequences you are trying to compare, and so it will be an n by n matrix which will tell you the connection between any two pair of sequences. So B and E are connected using a weight of 12, D and C are connected using a weight of 4, and so on and so forth, right? Now, how do you get the weight of these edges? So if you have a weighted tree and where you have the weight of this edge, in a tree, there's a unique path between any two nodes, right? So basically, if you want to move from E to B, there is one unique path, right? So you move from E to here, then here, then here, then here, right? Now, similarly, if you want to move from E to C, so you move here, 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 and here. So there are unique paths between any two nodes, specifically for phylogenetic trees. In other graphs, it is not the case, right? You can have multiple multiple routes from one node to another, right? But in this case, you have only one unique path. And if you consider these edges, while you are moving between these two set of nodes, then the distance between those two set of nodes is just the sum of the branches of between these two nodes. So for example, you're moving from E to B, what will be the weight, right? So you move from E to here, so this weight is 5, here 5 plus 3, 8, then you move from here to here, 8 plus 3, 11, and then you move from here to here, which is, you add 1, which is 12. Now E and B will have a weight of 12, difference of 12. So if you go here, you see E and B, you have 12, right? So this is how distance between any two nodes in a phylogenetic tree is considered, right? So you just wait. So you'll have weights of on each of these edges, because again, if you remember phylogram and cladogram, in cladogram you'll have weights, right? So the length of the branch will denote the weight of the branch, right? So if you have these weights, you can calculate the distance between any two nodes or any two sequences in a tree, right? Okay, I'll not go into very details of this, but I just wanted to give you an idea that if you have an existing tree and uh, you want to add an element to the tree, it's not very straightforward, right? So a distance matrix can not always be visualized as a weighted tree. So, for example, if you want to add, so this is, just consider A, B, C is your tree. Now what you want to do is add D to it. But according to these weights, you cannot add D, because if you add D, the distances will not be preserved, right? Now, what you can do is basically, I'll just quickly browse through this. There's a lot of writing here. Don't get too confused. It's just it's not very difficult. Yeah. So if you have any four species, say for example A, B, C, D, and they're arranged in an uprooted tree, like something like this, A, B, C, and D. Uh, there is a central spine that connects this particular pair AB to CD, right? So this is that spine. So if you connect AB and CD, this is the spine. The test is basically to calculate three possible sums of distance. So ACBD. So you are moving from AC, then BD. So this is the one sum that you're calculating. ADBC. If you're moving from this, so this is one diagonal. So you have to consider this is a as a quadrilateral or a quadrangle, whatever you want to call it. So first is basically you're calculating the distance between the parallel sides. So A and C, B and D, right? This is the first, and you're summing it up. Second is you're calculating along the diagonal, right? So A and D and B and C, sum of the diagonals. And the final one is basically the shorter lengths, so AB and CD. So this is, you can consider this as a rectangle from which you're calculating these distances. Now, if for the data to fit a tree perfectly, the two largest sums must be equal and they must be larger than the third sum. So this is a theorem that works. There's a proof to it. I'll not go into the proof, but this is the theorem that it is. So the largest distances AC, so AC and BD should be equal to AD and BC. So so ACBD should be equal to AD and BC, which should be greater than equal to the third distance, which is AB and CD, right? Or DC, whatever. So this equality or inequality, whatever, this needs to hold for a particular uh, tree to fit a weighted matrix. So the weights, as I was saying, the weights and convert a weighted matrix into a tree. This particular condition needs to be satisfied. You cannot convert a data to a tree perfectly unless and until you have this. I'll not go into the detail of this, but like this is a theorem, right? So the property of this theorem is like, so basically this is called the additive theorem, right? So if a particular distance matrix is additive, it then only it can be precisely represented as a weighted tree or else not. And by additive, you mean fulfilling this particular condition. Okay. Uh, Yeah, so again, I'll quickly go through this because the next slides, I think those are more important for you guys than this. So, but like, I just wanted to give you a foundation on this. Okay, so in a tree, the path between any two species goes through internal nodes. For a matrix to be additive, you need this particular condition, which I just showed here. Now, for for just for example, assume that you have an additive matrix. Now you want to convert it to a tree. How do you do that? So first, you start to form a two-leaf tree with A and B, when A and B are any two elements. Uh, for I equal to 3 to n, so two elements have been added from 3 to the end element. So this is pseudocode, by the way. So from 3 to n, you take any vertex that is not there in the tree and consider the vertex that they are in the tree, and then you calculate this set of distances. And from step 1, you know that the position of the C and the length of the branch C. If C did not hit exactly a branching point, then add C and Z. So for example, in this case, C is between X and Y, so this is a branching point here. If not, you add another Z here, and then you add a third branch for C, right? Uh, yeah, and if you take an example, this is basically the additive matrix. Then you calculate. So you take any two. So you take U and V first. So this is the first step, and then you the distance is 10. That is fine. Now from the third element, you take you take any two vertices and you calculate the distances like this. So C, X and C, and X and U. So basically, what you're doing is you're assuming a position C in between X, U and V, and you are calculating the distance from the rest, so X and Y, to that position C. So that you're doing, and then you're calculating for X. First, you're calculating for X. This is X and C. This is U and C, and then you add the final. So this is one iteration, and the second iteration would be this to add Y. So first was X, then you add Y, and then you calculate what is the distance between Y and C and U and C. So here also you're calculating the distance between X and C and U and C. So what you're doing is you're just randomly inserting a point C here, and then you're calculating the distance from that point to one of the points that is already available and the point that is you are adding. So from that you can get something of a tree like this, which with branch and branch lengths. But again, this matrix needs to be additive, and for it to be additive, it has to follow this particular principle. Yeah. So in the real world, most of these matrices are not going to be additive. Almost all matrices are not additive. Uh, so finding a tree that minimizes the error optimizing the error is hard. So for heuristics, we use two different types of methods. One is the UPGMA, which you have already, I think, studied, and another is the neighborhood joining. We quickly go over this, right? So what is the idea? The basic idea is that you are clustering similar type of proteins together, and you're finding distances between two similar set of proteins. So it's a basic algorithm. You need a distance function and you need a clustering algorithm. These are the two things that you need. So the basic algorithm is basically you put, I don't know how many of you, just a quick survey, how many of you have uh, learned about clustering algorithms? Are any of you familiar with k-means clustering? Okay, okay. Okay, ठीक है. I'll try to explain. Okay. Uh, okay. So the idea is that you are given it's a clustering algorithm where you know the number of clusters that are possible from your data, uh, and you're trying to find the best possible set of clusters for that number. So for example, you want to divide your whole data set into three clusters. You know that you want to do it for three clusters. Your work is to find the best three clusters. So that is a very high level understanding of k-means clustering, where k is the number of clusters. Right? Here the algorithm is similar, right? So how do you do the k-means clustering algorithm or clustering algorithm like this, right? So what you do is first you consider only one single cluster. So all your data is present in one cluster, right? Then what you do is you initialize a forest T of any node trees. Uh, I'm not sure if I'm going to be able to explain this clustering here. So let's how do I explain. Okay, so you are given, say for example, forget about k-means and everything. Just think about what I'm trying to say, right? So you're given a whole set of data. Say for example, you're given a set of heights of hundred individuals. Now, how are you going to say that? How are you going to construct a tree from that? A similar phylogenetic tree, right? So the idea is that you club similar individuals together, take a branch, and then from that branch you take the third individual which is most similar to these two, then you add a branch to that, then you take a fourth individual which is most similar to these three and add a branch to that, and so on and so forth. So the idea is that you take, at first you take any two random set of elements, and then you find the distance between them, and then find what is the third element which is closest to these two elements. And then you iteratively want to do that, right? So basic idea of clustering is that you need to find two set of data points which are closest to each other. So for that, you need a distance function. So generally, in real life data sets, what you could use Euclidean distance. So if it's in a two-dimensional space, you use Euclidean distance. If it's but again, if you want some sophisticated distance measure, you can use entropy or something like that, which are not reversible and so on and so forth. I'm not going to those complications, but the idea is that you need a distance function to say if two data points are very close to each other or not. And if they are close to each other, you consider them as single cluster. And once you have considered them as single cluster, you consider them a single cluster is a single data point. Now you take another data point and try to find which of among all the rest of them are closest to your current cluster. And then you keep on iteratively adding that. So that is a general idea of clustering, right? So here also, you're doing a similar kind of thing. You put every element in one element cluster. Initialize a forest T of one node. Each tree corresponds to one cluster. Find the closest cluster C1 and C2 and merge them into C. As I was saying, find two similar data points using a distance function. You merge them into one data point. That is one cluster. That is a single data point now. Now you take another data point and try to find the distance between that, right? So that is what you're doing. Find two clusters C1 and C2 and merge them into C. Compute distance from C to all other clusters. Add a new vertex corresponding to C to the forest T and make corresponding nodes from C1 and C2 from this node. So if you have C1 and C2, C1 and C2 will be connected to a single node, and C3 will be connected as a branch to that node, right? Because C1 and C2 are the closest, then you have C3, which is closest to C1 and C2, not individually C1 or C2, right? So C1 and C2 is a cluster C. So C3 will be closest to cluster 3. So like that, right? So you do this iteratively to find the relationship between which pairs of data points are closest to each other, and you make a phylogenetic tree out of that. So this is a distance function that they define in terms of UPGMA, the algorithm that we use.
Sir, so in UPGMA, we were just looking at the mismatches, and then we were seeing the closest mismatches, and then
Yes, yes,
joining them together and forming a
yes,
cluster, and then we are forming a new
yes,
matrix to join with the other
yes,
thing. So
yes.
that's the same.
That's the same idea. That's a clustering algorithm, right? So UPGMA, at the heart of it, it's a clustering algorithm, right? It's not very fancy. It is a very simple algorithm which checks what is the similarity between or dissimilarity between two sets of proteins or two sets of sequences, and then based on that, you say that if like you merge them into one, and then you consider a third element which is closest or to this particular cluster, and then you do it iteratively. So yeah, UPGMA in its heart, it's a clustering algorithm. But yeah, we are going to talk about UPGMA here as well. So yeah, I'll discuss that. Okay, so this is the distance function that they use. You can see that this is an average. So if you have, so C1 and C2 are the set of clusters. Now it across individual elements of the cluster, you want to find the average distance between these two clusters. So for one, so if you take one element here, you calculate all the distances from all the elements in cluster 2. So this is one distance, this is one distance, this is one distance, and so on and so forth. So one point in one cluster versus all the points in the other one. So, and then you take an average of that. So this is the distance function that is used in UPGMA. So the idea is that to combine hierarchical clustering method to put weights on the edges, and you add a distance function like this, and we need to come up with a method of computing this branch length. Sorry for the spelling of branch. So the algorithm is it's kind of the same. Two clusters C1 and C2, which are the closest, and then you merge them into one. Then you compute average distance between cluster C to all the rest of the clusters and add a new vertex based on which cluster is the closest to cluster C. And then you continue to do this iteratively. And basically, for the edge weight, you assign the length, which is basically half the distance between two clusters, as the edge weight between two corresponding clusters. So the idea is simple. If I suggest you guys go and take a look at hierarchical clustering. Right. The algorithm between UPGMA is hierarchical clustering. I think if you understand hierarchical clustering, you'll understand UPGMA. It's not a very big deal, right? So if you are not familiar with hierarchical clustering, you can go and take a look. I think there are NPTEL courses also. If I'm not wrong, Professor Biplab Bose from IIT Guwahati has a course on data science for biologists or something like this. The name of the course. It's in YouTube. It's an NPTEL course. There you can find all the explanations of this set of algorithms, not UPGMA, but the hierarchical clustering, k-means clustering, different types of regressions, and those kind of things. So there, if you or any other source that you have, I'll suggest you study hierarchical clustering. That is, if you understand that, you understand the UPGMA algorithm. So it's almost the same. Okay. So the final thing is basically a neighbor joining. It is also a heuristic method like UPGMA. Here, the idea is that you construct a tree by iteratively combining nodes that are neighbors in the tree. So again, it is also a clustering algorithm. It's basically what you're doing is you're finding the nearest neighbors of a particular data point and you're clustering them together. So for this algorithm, I'll suggest you look at k means or k nearest neighbor algorithms, things like that. Those are going to be very similar to neighbor joining. Right? So your idea is to find a pair of neighboring vertices which are the closest. Uh, but like again, they are in some cases they can be the closest, but they are not neighbors. So for example, B and C, right? B and C are not neighbors because B and C do not have a similar. Uh, so B and C are not connected to each other is directly is what I'm trying to say. So they do not have a direct connection. A and D has a direct connection, but B and C does not. But the distance between B and C is lower than A and D. But here, what you're trying to do is find the neighbors, not the nearest clusters, right? So here, the idea is that you find the nearest neighbors, neighboring vertices, not necessarily the closest vertices. Um you find the neighboring vertices and then you club them as into a cluster, and then again you take them as a cluster, rest of the data points as individual data points, and then you find which is the nearest neighbor to this particular cluster, and then you do it iteratively, right? So it's a similar algorithm, but this the formula is like 1 by clusters minus 2, sum of the distances between two set of clusters. And you want to find a pair of clusters which actually minimize this function, which is basically if they are neighbors or not neighbors is going to it's going to keep them. I'm not going to go into details, but the idea is similar. It's a clustering algorithm, right? So yeah, there are some tricks that it uses to penalize certain distances. So it calculates a penalty for each node based on how far it is from everything else, right? So for that, it uses this particular thing, clusters minus 2, for the distance function. And instead of looking at the raw distance, this looks at the pair that minimize this function, this particular function, as I said, if you would want the raw distance to be small, but you'd also want the nodes to be isolated from the rest of the groups. Biologically, it means that it pulls the neighbors together even if they seem very far apart. So, for example, in this case, A and D seem very far apart, but they are the actual neighbors because they are directly connected. B and C look very close to each other. They are close in terms of evolutionary history, but they are not neighbors. Right. So here the idea is that you find the neighbors and you join them, form clusters. Again, find their neighbors, join them, form clusters, and go on to do this iteratively, and until you find until no data points are remaining. So yeah.
Sir, as B and C are separate branches, they are not very close to each other, right?
No, in terms of the distances in these branches, so this is one,
In
this
it,
is two. So in terms of the distance between B and C, it is not very far apart. It is actually the closest. It is 4. But these are not neighbors because these are not directly connected to each other as A and D are. So you would want to find the nearest neighbor, the nearest element which is a neighbor. right? So in this case, for C, the nearest element is B, that is fine, but these are not neighbors. For A, the nearest neighbor is B because they are connected, although they are like distance wise they are far apart, but they are the nearest neighbor. They are connected and they have the smallest distance between any neighbors in the data. So that is how you find it.
Okay. Okay.
Okay. Yeah. Any more questions? Because then we'll move on to the uh, assignment problems. Okay. If no questions, we'll move to the assignment problems. Let me. Uh, I don't think I can. Oh, I'll. Okay. Excuse me. Okay, so this particular software is basically a kind of software that measures entropy. Entropy is basically a distance function. It is nothing very complicated or anything, but it's a distance function. So it basically calculates distance. A score of 0 typically represents perfect conservation. Only one residue type is present, which is basically it takes a log of that value. So log 1 is going to be 0, and negative scores indicate higher variability or entropy. So 0 is going to be more conserved than the negative value. The value that it gives is basically, so what it does is if there is only one residue present, sorry, if there is only one residue present, it will give 1. If say for example 5 out of 10 residues are present, so it will be half. If you take log of that, it will be negative. So 0 is more preferable to negative. So if I'm being clear, so if you have a score of 0 at position 3 and negative -0 0.6 at position 6, 3 will be more conserved because 0 means all the proteins have the same amino acid at position 3 versus all the proteins do not have the same amino acid at position 6. So 3 is more conserved than 6. Okay. Okay, this is just the formula I just showed you in the slide. So you just have to plug in the value 4 in this particular formula. So for unrooted trees, it's 2n minus 5 double factorial. For rooted trees, it is 2n minus 3 double factorial. So you just plug in n, and double factorial is basically you'll multiply with one alternative. So normal factorial would have been 3 times 2 times 1. Double factorial is 3 times 1. Same with here, normal factorial would have been 5 times 4 times 3 times 2 times 1. In this case, it's 5 times 3 times 1. So yeah, just use that formula. See a couple of B's and a A and a C. Okay. No, UPGMA is based on genetic distances. So genetic distance is how is genetic distance calculated? It's just the number of mismatches between two sequences, right? So yeah, it's based on mismatches, not matches. Or I saw a third option, hydrophobicity. Certainly not hydrophobicity. Can get confused between matches and mismatches, but the idea is that two sequences will the divergence or convergence of two sequences or how far apart they are is dependent on the number of mismatches they have, not the number of matches. Because yeah, and certainly not hydrophobicity. Okay, so for this, I think some of you might not be familiar with one letter amino acid codes. So I'm just going to put it there. I think this is fine. Yeah. Do not go with the molecular weight given here. Go with the molecular weight given here. Right. Do we have any answers?
Sir, give me a few minutes. It's a
Yeah, yeah,
lengthy calculation.
sure. Take your time. Yeah, the answer is correct. It's B. Uh, yeah, if you just add it, it comes to I think 880 Dalton, but you have to subtract. I think so the peptide bond is getting formed, right? So if there are seven bonds getting formed, sorry, seven amino acids present, there will be six peptide bonds between them, right? So there will be six water molecules that have been released. So each water molecule will have 18 Dalton mass, and so this will be 108 Dalton. If you subtract it from this, it's 772. Just the trick to keep in mind is the water molecule that are getting released. So yeah. again, I don't think you'll be able to make sense of this without the code. I personally have, I do not like always seem to remember the one letter code for the amino acids, so it's a problem for me. Technically, you should know the one letter code. Uh, yeah, technically you should know. I personally have issues remembering the one letter code, but yeah, in exam they might ask questions giving directly the one letter code. Even I'll say you should remember the three letter codes as well. Okay, there was A and a C as answer. Okay, the answer is A. So the idea is that you're looking for long hydrophobic amino acids that is spanning the layer. So again, I'm this region is present in A. Let me see. So, I think this particular region from VW till AY. Sorry, VW WY. Yeah, from this to this region. I think all are hydrophobic. So yeah. So what you so if there's a transmembrane helical segment, what you're trying to find is basically there will be a segment of the protein that is completely inside the bilayer. So basically, you need hydrophobic residues in that region of of your protein. So yeah, technically, you're looking for a long stretch of hydrophobic residues. So that is there in the in option A, which is this particular stretch. Yeah. So again, these questions are like previous year assignment questions. So these kind of questions might come in your exam. So I'll suggest uh, like pay attention to these questions and the type of questions that are there because similar questions might come in your exams. So practice this type of questions more. Okay. The answer is, I think, C, option C. It's basically what you're looking is alternative hydrophobic and polar or hydrophilic residues. So one hydrophilic, one hydrophobic, and one hydrophilic, a pattern like that. So I think this is the pattern that satisfies that. So yeah, again, single letter amino acid codes you should know because you can be given a question like this and you're going to need to find what amino acids these are. So probably it's better to know the codes to answer these kind of questions, and these kind of questions might come in exams as well. Okay. Yeah, yeah, that's correct. So basically, parentheses you have to write the elements that are clusters together. So A and B should be in parentheses. Is not connected to anything directly. So C outside, D and E again connected, F again disconnected. So option D is the correct one. Okay. Yeah, again, this is correct. So I've not discussed this, but this is a package known as phylogeny inference package, and it's a standard software for constructing phylogenetic trees. Again, the rest of them are, PDB is a database, FASTA is a format, BLAST is another different thing for sequence alignment altogether. So yeah, yeah, that's correct. So bootstrapping will tell you what is the confidence interval of the test statistic you are using. So plus minus how much is what the bootstrapping does. So basically, we look for 95% confidence interval. So yeah, that is the benchmark that we look for. But yeah, again, Anna, this you have all you need here. Okay. Uh, if it's one, okay, I two M 13 G is 14, R is negative 2, E is negative 0, A is 1, T is negative 1. I'm getting, I don't think C is the correct answer. Oh, I think, are you, oh, acha, I thought this was for this question, yeah. Okay, I'll give you another minute. Uh, Okay. I see a A. I think A is the correct answer. D is not certainly not the correct answer. I don't know how you got D. It's a simple addition. So basically, I for I it is 2, M it is 1. So if you're adding up, right? 1 plus 2, 3. For G it is 1 again, 4. For R it is -2, which brings it down to 2. For E it is -2 again. So 2 minus 2 is 0. And for A it is +1, T it is -1 again. +1 minus 1, 0. So 0 should be the answer, not -1. I don't know how you got -1. But yeah, it's just you add up. And since they were asked for average, you're supposed to divide by 7, but yeah, it's 0, so the answer is 0. Okay. Yeah, so that's all from my side. Uh, do you have any questions? The whole presentation is going to be uploaded. So I think you uh, have access to this particular sheet, right? So not this one. This particular sheet, I think you have access to, if I'm not wrong. Do you have?
Uh, yes, sir.
Yeah. So in this particular Google Drive, I upload all my uh, slides. So 3 week slide is there. The fourth week will also be uploaded. So you can get the questions from there. Okay. Oh, link to this particular. Okay, not this one. Um, so here all the like these sessions are recorded and put on YouTube. So the YouTube links are also there in this particular doc. Okay, then if there are no questions, I'll close this session. How to practice more questions? I think online you will find a lot of resources there are for like so these are like basic bioinformatics and these are like kind of so in current bioinformatics courses probably not so much of weightage is given on sequence alignment. It is given but in a different kind of a manner. Not so much importance is given on this protein databases and those kind of things. But so there are a lot of older material on the internet that you can find. So I personally, a lot of my slides have been taken from this particular. Just a second. Yeah. So this basically is the computational biology branch for NCBI, and they have a lot of courses. They have a lot of uh, books. There, I think practice questions are also there. So if you want, I think you can check this out, and like there are a lot of information there. So I don't think I'm not very familiar with like where you can practice questions from because yeah, I think you have to like do a search on the internet if I'm not. Uh, yeah,
Okay, sir.
yeah, I'm not very familiar with like so my expertise is a little bit on statistics, a little bit on genomics, a little bit on molecular biology, and it's an interdisciplinary thing. This kind of bioinformatics. So when I did bioinformatics in college, this was the bioinformatics that I studied, right? So, but right now, if you look at computational biology courses in, say, for example, MIT OCW, MIT OpenCourseWare, or Stanford courses, and courses like from, say, for example, Professor Manoli Skelis or people like those, right? So which are right now the pioneers of computational biology. So you won't like these topics probably won't be covered there. A little different things, a little bit more modern research topics will be covered there. So in my case, I had a book. If I'm not wrong, the name of the book is Lesk. The author name is Lesk, Arthur Lesk. It's, the name of the book is Bioinformatics, and the codes were written in Perl, if I remember correctly. But yeah, I think some older books like those you can use to practice a little bit more. Other than that, I think,
Sir, we
yeah,
have in our college, my teacher recommended Jin Xiong
okay,
book.
okay,
So
okay,
I
yeah,
am referencing that too.
yeah, yeah, sure, sure, you
But
can
my
definitely.
goal is to mainly clear the NPTEL certificate
So for
exam.
NPTEL, I don't think you need separate books. I think some book links have been given in by Sir in the course
Yeah.
page as well. So I think those would be enough. I don't think you need anything more than that. If you just want to like learn more, I think there are
Yes, sir.
other books available you can use. But yeah, I think some courses are also available if you want. So yeah, you have to search a little
Okay,
bit.
sir. Thank you, sir.
Okay. Any more questions? Okay, then I'll close the session. Thank you for joining. I'll see you next
Thank
week.
you so much.
Thank you.